The immune response is highly specific for each invader, and that's because the cells of the adaptive immune response have receptors that can differentiate friendly bacteria from potentially deadly pathogens from their unique parts, called antigens. The key cells of the adaptive immune response are the lymphocytes, the B and T cells. T cells develop in the thymus, where they undergo a process called VDJ rearrangement to generate a massively diverse set of T cell receptors, or TCRs. There are two types of T cells, which are identified based on molecules they express on their surface. Helper T cells express CD4 on their surface, and their main function is to support other immune cells. Cytotoxic T cells express CD8 on their surface, and their main function is to kill infected or cancerous cells. A T cell starts out naive, and then it gets primed or activated, at which point it differentiates into an effector T cell and proliferates. Activation of both helper and cytotoxic T cells requires two signals. After that, the cytokines present around the cell determine the type of T cell it will become. The first signal occurs when a T cell receptor binds to an antigen. Now, a T cell receptor can only recognize antigens that are peptides, rather than carbohydrates or lipids. And the T cell receptor also needs to have a peptide presented on a major histocompatibility complex, also known as an MHC. MHC molecules act like serving platters and present the antigen to T cells. There are two types of MHC molecules that work with the two types of T cells. MHC class I molecules present antigen to CD8 positive T cells, and MHC class II molecules present antigen to CD4 positive T cells. MHC class II molecules are found on the surface of an antigen presenting cell, like a macrophage or dendritic cells, while MHC class I molecules are found on all nucleated cells throughout the body. The antigen presented on the MHC molecule must be the right size and shape to bind strongly to the T cell receptor, which is part of the CD3 complex. The CD3 complex has 7 peptide chains: 1 alpha, 1 beta, 1 gamma, 1 delta, 2 epsilon, and 2 zeta chains. The antigen binding site of the T cell receptor is composed of the alpha chain and the beta chain, each of which have 2 domains, a variable domain and a constant domain. The variable regions of the alpha and beta chains of the CD3 complex of the T cell receptor bind the antigen, and the remainder of the CD3 complex binds to the MHC molecule. Whether it binds to MHC1 or MHC2 depends on if the T cell expresses CD4 or CD8. The constant domain has a short transmembrane segment and a short cytoplasmic segment. The transmembrane segment contains cysteine residues which allow 2 adjacent chains to form a disulfide bond, connecting the chains to one another. And the variable domain of the alpha and beta chain come together to form a single antigen binding site. This is different from the B cell receptor, which has 2 antigen binding sites. Also, unlike the B cell receptor, the T cell receptor is surface bound and cannot be secreted. Now, in addition to the T cell receptor, CD4 binds to the MHC class II molecule, and CD8 binds to the MHC class I molecule, and that helps securing the interaction between the T cell receptor and the MHC molecule. Second, the cytoplasmic portion of the alpha and beta chains of the T cell receptor are pretty short. As a result, the signal that the T cell receptor has successfully bound an antigen gets sent down other portions of the CD3 complex as well as the CD4 or CD8 molecules. The second signal required for T cell activation is called co-stimulation. And that's when a ligand that's on the surface of a T cell called CD28 binds to a ligand called B7 on the antigen-presenting cell. Antigen-presenting cells start expressing higher levels of B7 on their surface when there are inflammatory cytokines like interferon gamma, IL-1 beta, and TNF-alpha around. Now, if a T cell sees its antigen without co-stimulation, it becomes anergic, meaning that the cell won't get activated even if it sees the antigen in the future. This extra layer of security prevents T cells from automatically getting turned on. Just like how a cell phone needs to get unlocked to make a call, it prevents mistakes from happening. When a T cell and antigen presenting cell are interacting, the region where the T cell receptor is bound to MHC antigen and either CD4 or CD8 and B7 is bound to CD28 is referred to as the immune synapse. Once the T cell has received both signal 1 and signal 2, it has to send that signal down to the nucleus so that the cell knows it should respond. This is done using the CD3 peptide chains that have an intracellular component. In all, as we mentioned, there are 2 epsilon chains and 2 zeta chains, and 1 each of the alpha, beta, gamma, and delta chain. All of these, except for the alpha and beta chains, have at least one immune receptor tyrosine-based activation motif, or ITAM for short. The ITAM is a part of the peptide chain that includes 2 tyrosine amino acids perfectly spaced out by other amino acids between them. This is really important because both tyrosines need to be phosphorylated for the cell to get activated. Each gamma and delta have 1 ITAM region. The 2 epsilon chains each have 1, and the 2 zeta chains each have 3, a total of 10 ITAM regions altogether. The binding of CD4 or CD8 to the MHC molecule causes the kinase LCK, which is bound to the CD4 or CD8, to phosphorylate all of the ITAMs in the CD3 complex. Once that happens, another tyrosine kinase called ZAP70 comes along. Well, ZAP70 binds to the phosphorylated ITAMs on the zeta chains of the CD3 complex and becomes activated. Activated ZAP70 then phosphorylates the linker of activated T cells, or LAT, which is a transmembrane protein with a large cytoplasmic tail. ZAP70 also phosphorylates the protein SLP76, and LAT and SLP76 work together to trigger a chain of events that ultimately activates the major transcription factors NF-kappa B and NFAT. These transcription factors increase the expression of cytokines like IL-1, IL-2, IL-6, and TNF-alpha. They also cause upregulation of cell surface markers like BCL2, which provide a strong survival signal to the T cell so that it proliferates and differentiates. The activated T helper cell also begins making lots of cytokine IL-2 and upregulates its IL-2 alpha receptor. The IL-2 receptor has 3 protein components, alpha, beta, and gamma. A naive T cell only expresses the beta and gamma components of the IL-2 receptor, but these have low affinity for IL-2, in contrast to the alpha component of the IL-2 receptor, which has a high affinity for IL-2. Using just the beta and gamma components is like trying to eat an apple with only your lips and tongue. And upregulating the alpha component is like using your teeth to take a big bite of that juicy apple. Much more effective and satisfying. As a result, active T cells bind to IL-2 that they make, a form of autocrine stimulation, a cell stimulating itself. In response to the IL-2, the activated T cell starts to rapidly undergo cell division, a process called clonal expansion. If the cell is a CD4 T cell, it will also produce its own IL-2, allowing it to make and eat its own food. CD4 T cells also produce IL-2 for nearby CD8 T cells, which aren't as good at making it. Alright, as a quick recap. There are 2 types of T cells, helper T cells that express CD4 and cytotoxic T cells that express CD8. To activate a T cell, you need 2 signals. Signal 1 is the antigen in the MHC molecule, and signal 2 is co-stimulation, which is when CD28 on the T cell binds B7 on the antigen-presenting cell. This sets off a cascade of phosphorylation, which ends with the high-affinity IL-2 alpha receptor being expressed on the cell surface. This lets the T cell bind IL-2, like sinking teeth into an apple, which causes T cells to proliferate and differentiate.